Hello, everyone. I'd like to start by thanking the organizers of this meeting for inviting me to speak. It's my pleasure. Today, I will be talking about genetics and pathogenesis of kidney angiomyolipoma, the role of MITF and other transcription factors. So these studies really had their origin about 7 or 8 years ago when we had the hypothesis that other genetic events beyond biallelic loss of TSC1 or TSC2 would be important in renal angiomyolipoma development, particularly those tumors that were large enough to require some type of intervention. So we studied 23 angiomyolipomas that were available to us from various surgical sources and found that, in contrast to our hypothesis, that there was a very low rate of somatic mutation in angiomyolipoma. And several of them had no additional mutations at all beyond those occurring in TSC1 or TSC2. In addition, their genomes were stable, they were diploid, they had no regions of amplification or deletion. They did have copy-neutral LOH in many cases to eliminate the wild-type copy of either TSC1 or TSC2. So this led us to think further about why certain tumors develop in TSC, why some grow to be a large size and to be clinical problems, and why others did not. And so one important thing that we were aware of course, was that many transcription factors had been identified that played a role downstream of mTORC1 activation, including those involved in metabolite generation for anabolism, a hallmark feature of mTORC1 activation. And some of these are shown on this slide and listed to the right. They've been identified by various investigators, including those in the Sabatini lab and those working with my colleague, Brendan Manning. But another thing to consider here was that, in fact, a possible role for certain transcription factors, specifically MITF, had been identified many years ago in the pioneering studies by Franco Bonetti, in which he identified a consistent expression pattern among members of the PEComa family, including AML. In particular, a monoclonal antibody HMB45 with an unknown antigen at that time, but to which we now know binds to PMEL or SILV, was found to be expressed in the majority of PEComas. And this was followed up by another study by Andrew Fopi, in which he studied 26 PEComas, including AML, and found that, again, HMB45 was very consistently expressed. MITF and TFE3 were also expressed in a number of these tumors, and so was melon A, another protein downstream of MITF in melanocytes. So this led us to consider that perhaps transcription factors were very important in the generation of tumors that occurred downstream of TSC2 loss, as shown here. An important consideration here was that there are roughly 37 trillion cells in the human body, and a very conservative estimate would be that 1 in a million would have second hit loss of TSC1 or TSC2, so that there would be 37 million cells that would have the potential to develop into tumors in patients with TSC. Fortunately, this is not the case, suggesting that TSC2 loss does not have the same effect in all cells. And so in an unknown cell of origin, still to this day, that has the right chromatin state, I will define that further, that loss of TSC2 would lead to mTORC1 activation and lead to an increase in activity of certain transcription factors, including MITF. And I'd like to highlight that although increases in transcription factors downstream of mTORC1, that might happen in all cells, but to somewhat of a variable degree. But in addition, the expression pattern of the starting cell, the constellation of transcription factors expressed, might also be very important in determining whether or not mTORC1 activation leads to a tumor or leads to a cell that undergoes senescence or something else. I'd also like to note that this hypothesis is now 5 years old since I first presented this at a LAM conference. Now, one important thing to be aware of is that MITF is one member of a family of transcription factors called the MITF-TFE transcription factor family that includes TFE3, B, and C. And although classic sequence motifs have been defined for both MITF, based upon analyses in melanocytes, and for TFEB, based upon studies looking at lysosomal biogenesis, in fact, these motif DNA sequence patterns are very similar if you look at them, such that it is quite possible that these transcription factors bind to the same motif in many circuits. It might be MITF, but it might also be these other transcription factors. So another thing that's important to consider is what is the relative level of expression of these transcription factors in AML and other tumors. So here are RNA-seq data for MITF, TFE3, and TFEB. For AML, shown to the far left, showing that MITF is the highest, with TFE3 being second and TFEB being quite low in comparison. Whereas for other tumor types, including malignant pecomas and SEGA, a TSC brain tumor, this MITF was not so highly expressed, but TFE3 was the transcription factor member of this family that was highest. I'd also like to note that this is based upon RNA-seq data, mRNA levels, and certainly is very valuable data, but does not directly correlate necessarily with protein levels. So it could be that TFEB expression is actually much higher than what one would infer from looking at this diagram. So to understand this further, we have pursued chromatin studies on angiomyolipoma and other samples that have been available to us. And so what we have studied is a chromatin mark that is a modification of histone 3. It's called H3K27 acetyl. And this chromatin mark can be assessed genome-wide through a process called H3K27 acetyl ChIP-seq. And so this identifies all the regions in the genome where there is significant binding to this specific modified histone. And it's known that this histone occurs in regions of open chromatin, not in regions of closed chromatin that is not available for TF binding. And so these open regions typically are near transcriptional start sites, as seen here for CTSK, but can also be seen in enhancer regions that are remote from TSSs. So this slide shows that for 5 AMLs, the H3K marks were very high and intense here in the CTSK region and were completely absent in normal kidney as a control. Now it's possible to quantify the level of H3K27 marks in the transcriptional start region for all genes across the genome and to compare them to a set of normal tissues, which we did in this case, including 10 renal cell carcinomas, and to assess what is the ratio of marking with H3K27 compared to, again, normal tissues. So you can see that this starts with a few genes that are very highly marked relative to control and then steadily plateaus down, extending out through all genes, although this graph stops at about 7,200. And if we look at those genes that are the most highly marked, we see that these match up very well with what could be considered marker genes for AML, including ABCB5, CTSK, PPAR gamma, mLANA, GPNMB, and also MITF, which comes in at 82 or 83 on this scale and is much, much higher than what we see for TFE3 or TFEB, which are relatively lightly marked, comparable to many of the normal tissues. So we then went on to examine this in greater detail and saw that indeed MITF was expressed in angiomyolipoma and had a nuclear localization in the majority, and that by this same H3K27 acetyl ChIP-seq, that there were very high markings in the region of MITFA, consistent with the regulatory potential data I just showed you. And that importantly, this region of marking was corresponded to the MITF A1 isoform and not the MITF M isoform, which is shown here, and which was marked in this melanoma cell line, SKMEL30. Looking at this at higher power, we can again see that for these 3 AMLs, there's relatively high marks for in the region of the MITF-A transcription start site, and much weaker in the melanoma cell line and a normal kidney. And again, there's this valley here between certain marks, consistent with the possibility that transcription factors would bind there. So this led us then to pursue, assess whether or not MITF was truly a critical gene for growth and development of AML. And so we used the AML 621-101 cell line derived from an AML, and both knocked down MITF expression using CRISPR-Cas9, generating KO lines, and overexpressed MITF-A as well, generating OE lines. Validation of these lines is shown in figures B and C at the top of this. And most important is that the OE cell lines grew faster, cells per dish here, compared to the empty vector transfected 621 cells. And that control line was also much faster in growth compared to the MITF knockout line. This is also seen in a low dilution clonal growth assay and in assays of migration through a transwell membrane. This work is now published, and Oncogene majority was done by Masa Zarai, a fellow in my group. Most importantly, these MITF, KO, and OE cell lines had opposite growth phenotypes in vivo. So when these 3 different flavors of 621 cells were injected into nude mice, the MITF knockout cells did not form tumor nodules even after 2 months of observation, whereas the control lines developed tumors beginning at about two months, and the overexpression lines began to develop tumors at about a month after injection. So again, strongly suggesting that MITF had important growth promoting properties in angiomyolipoma. However, it's still unclear whether it's all of MITF, A, TFE3, and TFE-B that contribute to TSC tumor development. And something that Clemens Probst pointed out in this sequence alignment plot is that, in fact, MITF-A, TFE3, and TFE-B have very similar sequences with many regions of homology, including the basic DNA binding domain, the helix-loop helix domain, and the leucine zipper domain that enables dimerization. So although MITF is important, it could be that TFE3 and TFE-B also contribute. So to try to dissect this further, it would have been ideal to have an authentic animal model of lamb, but such does not exist. But instead, there is an experiment of nature which we can use to examine this. So there's a subtype of renal cell carcinoma called papillary RCC, and about 3% of PRCC have TFE3 translocations gene fusions that put TFE3 into the nucleus and make it transcriptionally active. And similarly, there's about 1% of PRCC have TFE-B translocations gene fusions that have the same effect. And since TCGA RNA-seq data for 274 PRCC are available, this gives us a total of 8 tumors that have TFE3 fusions and 2 that have TFE-B fusions among this data set that can be compared to the remainder without those gene fusions. So if we consider those genes whose expression is up 4-fold compared with controls at a Q value less than 0.1, there is significant overlap among these 3 types of tumors, with 24 genes being seen in common to all 3. And so, in fact, in all 3 types of these tumors, they all appear to be drivers of the expression of many classic TSC marker genes, including ABCB5, as shown here, markedly increased in AML and in PEComa compared to leiomyosarcoma, increased a bit in SEGA, and in astrocytoma, this is completely flat, and very significantly increased in TFE3 fusions and TFE-B fusions, suggesting that all 3 of these transcription factors could bind to and lead to high expression of ABCB5. So this is also true for TRIM3, for CTSK, and for GPNMB. High level expression is seen in all three of these tumors, including AML. I'm not going to take the time to go through all of these, but it is not true for all classic TSC marker genes. So for example, PMEL and MLANA, shown at the top, have high expression in AML, but minimally no increase really at all in the TFE3 fusion or TFEB fusions. With one sole example, one of the TFEB fusions had relatively high expression. And this is also true for VEGF-D, very high in angiomyolipoma, not up at all in the two fusions, and also for estrogen receptor, where the increase in expression is not as dramatic, although it is still up, but again, really a minimal change for the fusions. And again, this is in without marked change in expression of these genes in these different tumor types, but rather the fusion mutations, for the most part, move the TFE3 or TFEB to the nucleus and make them active with respect to driving transcription. The TFEB fusions are exceptions to this, such that they do lead to increased expression as well as activation of transcriptional activity. So to understand this further, we have also performed MITF ChIP-seq studies on a limited number of samples and are working to do more. But we have seen that MITF fits precisely in the valleys between these H3K27 acetyl marks that would be predicted in the transcriptional start site promoter region of CTSK, as seen here, not seen for MITF in the melanoma cell line. And in addition, that MITF-A binds to its own promoter to apparently drive expression of the MITF-A1 isoform. So this makes it a master transcription factor that is serving to help create the identity of the angiomyolipoma cell. And again, it falls precisely in this valley between the H3K27 acetyl peaks that we see in this region in those two AMLs. In addition, quite remarkably, MITF-A appears to bind to the promoter of both TFE3, shown here, again in this valley region between these peaks of H3K27 acetyl, and also for TFEB. It's a weaker signal, but still there is some signal here compared to other regions nearby where there's no signal at all for MITF ChIP-seq. And again, there is a pattern of H3K27 acetyl that is consistent with this gene being regulated by transcription factors nearby. So in conclusion, MITF, TFE3, TFE family members are relatively highly variably expressed in TSC tumor types with nuclear localization. MITF is the most highly expressed in AML, while in SEGA and PEComa, this TFE3 is highest, though expression does vary from tumor to tumor. And again, this is based upon RNA-seq data, and protein data may be quite different. 621 cells are significantly diverged from angiomyolipoma and MITF expression, but still dependent on MITF for growth, and overexpression enhances growth in vitro and in vivo. It is likely that multiple downstream effects from high expression and activity of these TFs contributes to tumor growth, including CYR61 and angiomyolipoma, something which I don't have time to talk about today. MITF binds to its own promoter in AML, indicating that it is a master transcription factor driving its own expression, and likely that of other TFs that are critical for AML growth, including TFE3 and TFEB. Other master transcription factors for AML growth are yet to be defined. Other things that we're working on include NR2F2 and MICE2 and AML. And in addition, I don't have time to cover this, but I would refer you to talks by Dario Lemos and Yan Tang on an iPSC AML model and single cell studies of AML, which also support the importance of MITF expression in AML development. So to end, I'd like to modify our previous model with a bit more details, although there's still considerable more work to be done. Still, TSC2 loss occurs in an uncertain cell of origin in the right chromatin state. This leads to mTORC1 activation, leads to, at least in AML, relatively high expression of MITFA, which binds to its own promoter and leads to a feed-forward effect in terms of enhancing MITF expression, and potentially, apparently, driving expression of TFE3 and TFEB, and likely, very likely, contributed to by other transcription factors yet to be elucidated. I'd like to acknowledge that through joint meetings that we've had for many years now with Lisa Henske's group, that I think there's been cross-fertilization of these studies with work that Nico has performed in Lisa's lab that are more biochemical in nature and looking at modification and nuclear localization of these TFs. I think this has been beneficial for both of our groups. And finally, I'd like to acknowledge the people whose work I have shown you today, particularly Masa, Crinio, Clemens, and these three more senior individuals down here who are not members of my lab, but have provided important insight and suggestions throughout these studies on chromatin and transcription factors. Thank you.